Hello everyone, and welcome to the webinar on custom technology for bioprocesses, hosted by IRNAS, based in Slovenia. We are very excited to host this webinar today, as custom technology is a topic that's been more and more explored in the recent past, and we are very excited to hear what all of our exciting panelists have to say about it today. As well as all attendees, please do get engaged. Feel free to post questions, post comments, challenge each other's ideas, because this is where the magic happens and the value is created. Just a bit of practicalities before we dig in. There's a Q&A section. There's a button at the bottom of your screens that you can open up and post all the questions, we promise to attempt to answer as many of them during the webinar. At the end of the webinar, you're all going to receive the link to the recording, which you can then share amongst your teams. Alongside with the contact details of all the panelists, who you can then contact directly in case you're interested in more details of their work. Without further ado, I'm going to present our panelists for today. First one on the list is Dr. Ivana Gadjanski. She's an associate research professor and assistant director for science at the BioSense Institute. Hello, Ivana.
Hi.
Next to her is Dr. Saam, who is the CEO of OSPIN, of OSPIN company based in Germany. Hello, Jan.
Hi there.
And our third panelist is Scott Allan, who is currently pursuing a PhD at the University of Bath, based in the UK, and is also a New Harvest Research Fellow. Hello, Scott.
Hi there, everybody.
So my name is Luka Banović. I'm an engineering project manager at IRNAS, and I'm going to be your moderator for today. So IRNAS is an innovation lab for applied custom technological solutions based in Maribor, Slovenia. IRNAS has existed for 5 years and has been developing custom technological solutions in several different fields, from free space optics to IoT, nature conservation, medical devices, industrial IoT solutions, animal trackers, etc. And about 2 years ago, we also got involved in developing of technological solutions for the field of biology and related fields. We are an engineering company and complete product development service provider, covering electronics engineering, software development, mechanical engineering, and implementing all that in our in-house manufacturing workshop, where we also produce and test all our products for different applications, and then at the end do the design transfer into mass production. We've been exploring the idea of custom scientific technology for the past year more actively, because we try to translate and apply principles that we've learned and explored in other fields of technology onto the field of biology and scientific research. We do that through formation of scientific technological partnerships, where we play the role of the technology provider throughout the lasting of the whole project. In essence, this means we work closely together with a researcher or a group of researchers, modifying, developing, and customizing their laboratory equipment, through iterative development process, and with the goal to maximize the value of the technology they use at all times of their their work. Currently, we have two major running projects related to this concept. One is a modular perfusion bioreactor platform that we are developing together with New Harvest, and the other one is our own spin-off project called VitaPrint, which is a family of custom CNC solutions very often applied in a scientific environment. So the former, the modular perfusion bioreactor platform that we are developing with New Harvest, is a system of cell incubator and perfusion trace working together as a whole unit. It's microcontroller operated through a web interface and is highly configurable depending on the final application. It's equipped with different sensors, with different modules such as heaters, temperature sensors, CO2 monitors, obviously the CO2 source. So it's a completely standalone operation unit that can either be that it either can perform tasks autonomously or it can be manually operated. Its complementary part is perfusion tray. It's again a microcontroller-based syringe pump system with bubble traps and tissue platforms for tissue growth. That is compatible to the incubator itself and works together with all the sensors and heat distribution and other regulation systems in the system. We use this platform as a baseline for customization. This means that once this gets functional and released, we're going to be using it to build on it and develop custom solutions for different use cases. The idea is to distribute this platform in various use cases and extract features and kind of necessary parameters to develop it further and optimize it for each of the use cases specifically. Currently, the system is under biocompatibility validation here at the local medical faculty, and the results of that are going to be available in a few weeks' time. Onto the VitaPrint custom CNC solutions. VitaPrint is a family of CNC solutions that has implications in different environments, but focusing it only onto the scientific field. We are developing CNC solutions for 3D biofabrication, manufacturing of perfusible structures, material property analysis, scaffold fabrication, liquid dispensing solutions, etc. It is a platform that has been under conception for about 3 years. That is currently at the stage where it can be put in almost any environment that we've faced to date, and can be easily integrated with the other peripheral systems that it's supposed to work with. This is a very brief summary about what we do and how we are involved in the field of custom technology and bioreactor technology. Now I would just like to invite you to kind of direct your thoughts towards what I would like to see today's discussion to be about. So we are interested in everyone's perspectives on what you see as advantages or disadvantages of custom technology in science. Please share your experience, challenge each other's ideas, think about where you see a place for that in the future, and hopefully we all go home with some new knowledge and some new insights from this hour's session. Thank you very much for listening, and I would now hand over to Ivana as our first presenter.
Okay, thank you, Luka. Just to share the screen. Okay. So, do you see my slides?
Mm-hmm.
Okay, great. So thanks again, Luka and IRNAS, the others also from the team. We've been collaborating also before, and I hope to continue in the future. This is a very interesting topic, and I'd like to tell you more about how we customize different components of hardware for our research on technology that can be used for cultivated meat. I'm going to present some data from the two projects that we have that are funded by the Good Food Institute, RealSense 1 and RealSense 2. And there is a link on the lower right corner, realsense.rs, where you can see more details on the project. So I'm not going to go that much now for the sake of time. So to tell you in short about BioSense Institute, or the full name Institute for Information Technologies in Biosystems, it's part of the University of Novi Sad in Serbia, and it's a pretty young institution, but actually since 2013. But we are quite, I can say, successful in getting different projects, primarily from the Horizon 2020, and now also Good Food Institute. And I'm mentioning this about the projects because it also has to do with this specific topic. The organization of the institute is, we have three research centers: Center for Information Technologies, for Sensing Technologies, and for Biosystems. And we have these two development centers for innovation and business development and for product development. And this structure actually reflects components of the whole BioSense vision of the agriculture of the future. So the Center for IT deals with the remote sensing, data analytics, so big data, artificial intelligence. We can we have quite good success in yield prediction, for example. The other center for sensing technologies, where we have the groups for nano and microelectronics, robotics, IoT, works on actual development of the sensors. And we have the third center for biosystems that works on different biosystem research, biodiversity. We have a group for bioarchaeology, and the new group is being formed that will be focusing on cellular agriculture. And as I mentioned, we have a number of quite large projects, and the flagship project is this one, Antares, which is done in collaboration with Wageningen University and Research from the Netherlands. And it's huge project, 7 years long. We are now in the third and a half, so basically half of the project. And there's going to be investment in infrastructure. So we have a new building being built, and the whole point is to transform the Institute BioSense to the European Center of Excellence for Sustainable Agriculture. And the plan is to have in this new building also the lab that will be dedicated to tissue engineering, cellular agriculture. So that's like very good development, I would say, from our side. I'm not going to go into general topics on what is cultured meat. I suppose Scott will also talk about that later. I just want to list these main challenges, although they're also quite well covered in the blog that was introduction. So Luka and Boštjan, that was really great text on the blog. Basically, what we're trying to do with our projects, RealSense from Good Food Institute, we're trying to use sensors to optimize the bioprocess of cell proliferation. So this is the mind map made by the Good Food Institute. It's pretty old now when you think about it. It's from 2017, when they still called the cultivated meat, they called it clean meat. Now they're really strong on using cultivated meat as a terminology. And you can see, but it is old, but it's actually, unfortunately, still pretty well valid, because the number of these different topics are not really solved. So cell lines issue, that's still a big issue. Scaffolding as well for the whole cuts. I'm not going to go into supply chain and distribution because we don't even have yet the commercial cultivated meat product. Cell culture media, maybe there we can say there's been the most progress, and bioreactors, and you see sensors and inline monitoring. So basically we in BioSense, since you see we have a strong background in sensing technologies, we focused on this. And just to repeat again, out of these two phases in the process of meat cultivation, we focus for now on cell proliferation. So our sensors are being made with the idea to be integrated into the steered tank bioreactors. So we're not working yet on the perfusion bioreactors and the second phase, tissue perfusion and maturation. So the first RealSense 1 project started in 2019 and it's ongoing. And it's probably going to be prolonged because of the whole situation with COVID-19, and we cannot really go into the lab, and it's, I suppose, the same for everybody, but it's really annoying. And the point of RealSense 1 is to make prototypes of sensors, low-cost sensors. So we're trying to optimize the cost from the beginning. The sensors for biomass and for different nutrients and metabolites. And the idea is to use this improved monitoring of cell culture to potentially reduce the cost, because if you can monitor the level of different nutrients in the media, potentially you can also okay, you have to have other components of the technology developed, but you can also potentially recycle the media, which would definitely influence the costs. They could be lowered. And the point with RealSense 1 is that we focused on the first lab on a chip approach. So we're using microfluidics. Because we wanted to, again, optimize as much as possible. We wanted to have just laminar flow and small volume, and where we test the first iterations of the sensors. And then in RealSense 2, which is the project that started as a follow-up, it started this year. So they're basically going in parallel. My colleague Vasa Radonic is leading RealSense 2. And in RealSense 2, we basically want to scale up, meaning to go from the microfluidics into the benchtop bioreactors. And in order to do this, we are going to use also the modeling, computational fluid dynamics, and to first model the gradients of different nutrients and the fluid flow in different specific model of the bioreactor. And that's why I was mentioning to Jan earlier that it would be good, very good for us to partner, to collaborate with the company that actually makes bioreactors, so that we can have the actual model of the bioreactor to use for modeling. And yeah, why to go into microfluidics? Obviously, I mentioned already, it's much more time and cost effective. And also it's quite a usual step in bioprocess development to first figure out as many parameters as you can in your lab on a chip device, and then move up in volume. And this is where the customization of technology was actually necessary for us, because we needed the specific design of the microfluidic chip, and I mean, we wanted to integrate the sensor in it, and we first wanted to, we didn't know in the beginning exactly which design of the sensor it will be, so we had to basically co-develop both the device, microfluidic device, and the sensor itself. And just to quickly go over this, so I'm going to show most of the data concerning the impedance measurement and the impedimetric sensor. So we are combining actually two ways of assessing biomass, but for this point now, for developing the sensor, the impedance measurement is the most relevant. And this is how we started. And I think we actually have on the webinar also two PhD students from BioSense, and Ivana is actually working, another Ivana, she's working on this fabrication of the custom bioreactor. We call it like this. It's a microfluidic chip. So we try different materials. We try different methods. As I said, we always try to keep it as cost effective, as low cost as possible. And we were testing then these different, this is a very simple design, and we tested just how the cells like it, and they actually did like it. I have to say that we are using the fibroblasts, and these are human fibroblasts, so these are just model cells. We are using them because they are adherent cells. And the bovine myoblasts, for example, are also adherent cells, but this is much easier for us right now to just for experimenting, for testing purposes, we're using fibroblasts. But once we figure out the whole design, we can establish we can actually start testing with the really relevant cells. This is now the next step of the, we can say, customization. So we changed the design a bit because we also wanted to include other types of sensors just for these basic parameters like O2, CO2, pH, temperature. And I'm not going to go now into details, but if you have questions, we can go through it there. And also the part which we also customize is the supporting electronics. And we first used this impedance analyzer, which was not actually intended for something like this to be used in this kind of micro bioreactor. So we optimized it using different microcontrollers, and now it's actually, it can fit into the whole incubator. This is another thing which we do it ourselves. So we're using, I just put here the microbiology incubator, which we use not for this, but for other studies, and the CO2 incubator with integrated, well, with the inlets for the sensors. And this CO2 incubator was made using as basic design the Andrew Pelling design, very nice one, and also not expensive. And this was made by our maker in residence. I have to say that's very useful to have, Jovan Lazarevic. And just to say, to mention about the sensors for nutrients, so which nutrients, like glucose and different amino acids, and the metabolic waste products, like lactate or ammonia. And this is something that I'm not going to go into details because it's actually more chemistry. It's not now hardware production, but we're making these nanoconstructs with the enzyme on top of it. And okay, you can read, but I don't know how much time I actually have. Maybe I overdid it already. So this is something we are still working on. I mean, we made these nanoconstructs and we are testing them. We have some problems with actually attaching them to the surface in the chip. So we're figuring out this. And in parallel, we are also working on this, as I said, scalability. So to move from the microfluidics, where things are kind of easy, at least when you think about the fluid flow, it's laminar. But now if we go into these steered bioreactors, things get much more complicated. And we partnered with Brown University, the Harris lab. They will do the modeling. And we are still figuring out actually what is the best way to let's say, integrate these sensors. For the impedance, we have a bit of a problem because the cells in steer tank bioreactor, these adherent cells, they need to be on microcarriers. And then these sensors for impedance, it's a bit tricky where they should be. So we'll see about that. And these sensors for the nutrients and the metabolites, most probably we have to think about some kind of a outlet where only media, a sample of media would go, and then this sensor for nutrients would be in there. but that's still work in progress. Any ideas, suggestions are more than welcome. And just for the end, I'd like to introduce this great team that works on both projects. As I mentioned, I think Mila and Ivana are here on this webinar, and Vasa, Goran, Nicola, Nicolina, they all, and Vesna. Vesna is our molecular biologist, and she works on cell culture. And Jovan, I really have to say, he's really a maker. So I think every lab should have at least one. So that's it for me, and these are my contact details, so feel free to get in touch also after the webinar. I hope I wasn't too much over time that you planned, Luka.
Thank you, Ivana. No worries. It was very, very interesting hearing from you and your work. I already have a few questions for you, but let's wait with that till the end. And I'm sure some people on our list of participants also have them. For everyone that have joined during Ivana's presentation, I just want to wish you a nice welcome, and sit back, enjoy throughout all of our other presentations. In case you've missed anything, there will be a recording available. Now, let's hand it over to Jan.
All right. Well, I'm glad to be here in this interesting session. I'm going to share a few thoughts with you today about this interesting topic about custom bioprocess technology, especially in the context of cellular agriculture. I'm the CEO of OSPIN. OSPIN is a company that develops a modular platform for bioprocess automation. We basically develop bioreactors based on certain bioprocessing modules, which then can be remote controlled through a web application. So you can build up your device, or we can build it up for you out of different sensors and actuators and controllers. And we are not focusing on a certain bioreactor type, which will come clear in a moment, but we're basically offering any bioprocessing solution. I'm not going to focus too much about what we are doing, what we are selling. Just check out our website. I'm going to share a few thoughts with you now that I hope might spur the discussion a little bit. So if you think about cellular agriculture, then most people immediately have these pictures in mind, like these stock images of large steel tanks stirred bioreactors, because everybody thinks like, yeah, we must scale, then everything's big. And the only bioreactor that sort of everyone knows is the stirred tank. But I am questioning this, and I'm saying, do you really think one tool is enough? And I make an argument here. So cellular agriculture is not a homogeneous field. Think about it. There are people that are trying to grow pork. There are people that are trying to grow beef. There are people trying to grow fish, or some other more exotic species like kangaroo, whatever. In cellular agriculture, you also have other products, animal products that are being produced, for instance, milk, and there's many more to come. And you then also have to distinguish between the different types of cells that they want to grow. For instance, some want to grow the muscle cells, some want to grow fat cells, some want to grow everything together. And these cells, they grow all under very different biological conditions. Some grow in suspension, some need a surface, some only grow in aggregates very well, and some actually need to grow in different conditions throughout the process. And each cell line is actually also very different. Some have GMOs, they can make them really resistant. Some say, no, we need to use the myocytes, but then they are very fragile. And the cells differentiate themselves by how many doublings do they actually have. And some of them are very shear stress sensitive and some are not. And do you really think that this very, very diverse field you can solve this all with one bioreactor type, which seemingly many people think. I say you can't. You actually need many different devices. So the question actually comes down to how to develop a bioprocess. And let's just go roughly over one example here. If you want to grow cells like muscle cells from myocytes, then you first have to expand the cells and you have to keep them in some state of stemness so that they really still like to expand and divide. At some point, you want to turn them into muscle cells and actually need to differentiate them further and mature them and really make sure that they build up this sarcomeric protein content. And you need different process steps for that, and you need different devices for that, most likely, or one device that can host these different process steps. And a priori, it is not at all clear how your bioprocess will look like exactly, because you need an automated technology that can be optimized, that can adapt to the bioprocess and later scales up with you. So the question is, how can you actually achieve that? And what we think is that we need something like a Lego system for bioprocessing. What you see on the left is a bunch of bioprocessing modules. It can be something very common, like a pump. Obviously, you need a lot of pumps in a bioreactor system. You need sensors like a glucose sensor, lactate sensor, pH sensor, obviously, temperature sensors, and then you need something like a gas supply. You need reservoirs, you need tubings, and all these things we treat them as modules that you can put together in different ways, creating an individual setup that makes up your bioreactor. So these building blocks can be combined freely, and then when you plan that with us, you can basically build a device for you. This device can be very small, can be benchtop, but it can also later grow. I would like to just share an example how you could use this modular approach to think about a bioreactor for, oops, something's automatically advancing here, to think about a bioreactor for the cultured meat field. I'm just going to go through it real quick. I'm not sure if you can see my mouse here, but in the middle you sort of see this quadratic circle, which is the main perfusion cycle, and where you have some kind of chamber in which you grow the cells. I'm getting to it, what kind of chamber that might be. You would want to have something like a measurement loop where you constantly take out some sample medium, measure things like ammonia, pH, oxygen, lactate, and bring it back into your cycle. Ivana has already mentioned that medium recycling is very important in the field of cultured meat. So you need to come up with a way how to make your medium last longer. And obviously there's some filtering approach that you need to apply. Without going into any detail, you would need to have such a system where you can do that. And then another thing that is very important is feeding. Obviously you need the basic medium, but the cells are not going to use everything that's in that medium in the same speed. So you might want to feed glucose and certain amino acids while the process is running. And with this sketch that I did here, I already showed that that you need these modules to create such a setup. And obviously, anybody of you who is into that field would immediately say, no, but I need the sensor somewhere else, and I need another pump, and I need another module. So this is really the case where you need to have a system that allows you to construct custom bioprocessing equipment. So I was leaving one thing out. I said just some cell growth chamber, and here again comes why we need very different systems. This cell growth chamber, in some cases, the stir tank might just be the right tool. In other cases, it's a rocking motion platform, which has the advantage of having much lower shear stress. If you really need adherent cells, then you might be better off with the hollow fiber system, or you want to have aggregates in a shaker. That really depends. There are so many different bioreactor types out there. And again, this is an argument for modular bioprocessing and for having custom-made bioprocessing devices. So that's my last slide here. We really believe that what you need in such a growing field where so many things are not yet defined, you don't only take something out of the shelf that was made for the biomedical field and then try to use it. You really need to work together with your bioreactor manufacturer and create a bioprocess together in an iterative process and develop your bioprocessing technology along the way. So that's it already from my side. I hope I could give you some insights about my thoughts about this field, and I'm giving back the control to Scott.
Thank you, Jan, for this. This is exactly the added value of people like yourself that can provide it on this topic. So thank you very much for this. I also have some questions ready for you already, so can't wait to be able to pose them. But for now, over to Scott.
So, hi everybody. Just wanted to again thank IRNAS for the invitation to speak to you all today. My name is Scott Allan, and I'm a PhD student at the University of Bath in the Department of Chemical Engineering. I'm a New Harvest Research Fellow, and I'm working with Dr. Marianne Ellis and Dr. Paul de Bank. And my research is focusing on bioreactor design for scaling up cultured meat production. And within that, I'm working specifically on hollow fiber bioreactors and focusing on defining some of the fundamentals. But today I thought I would give you a bit of an overview of cultured meat and bioreactor design, and then I'll talk to you about some of the custom technology that we use within our labs, which is actually primarily centered around in-house fabrication of different scaffolds. So just to give you a brief overview, this is very much a simplified schematic of what cultured meat is. I wasn't planning on spending too long on the slide. I was under the assumption that either Jan or Ivana would speak about this a bit more. But this very much gives a general overview of what we can expect from a cultured meat process. And my focus is going to be on the bioreactor and scaffold side of things. But first, I want to just briefly talk about why we need cultured meat. If you think about our traditional meat production system, I think everybody will agree that it's not what we would typically call ideal. And what I mean by that is if you imagine ideality or a perfect food production system in terms of its production costs and price, and whether it's sustainable in terms of its environmental impact, the animal welfare side of things, and also whether it's got a high productivity or production capacity, i.e. can it feed the growing population. I'm sure we will all agree that there are definitely areas for improvements within our traditional meat production methods. So what we can think of is essentially how can we create a more ideal system. So we could be looking at meat analogs or meat substitutes, or we could be trying to find a different production method for our current meat products. So over here, I've got another S-curve for cultured meat, and currently it's still hypothetical because there are no products commercially available. However, the way I've drawn it currently is it is quite possible that at the stage of initial commercialization, it is not necessarily as ideal as our current meat production system on all fronts, whether this be in terms of industrial energy requirements or in terms of the actual cost of production and consequently price parity. Basically, it shows us that there is definitely a need for more players within the field so that we can all contribute to the advancement of this technology. And as more funding and more people start researching this, we can expect to reach that acceleration phase and see exponential growth and a move towards an ideal food production system. Now, some of the areas that we actually need to focus our research on can be broken down into 4 different technical challenges. So the cell culture media, cell line, scaffold, and bioreactor and process design. However, even if you are working on one of these areas, it's important to remember that they are all interlinked, and any decision you make with respect to one area will have an impact on the others. So as an example, although my focus is on bioreactor and process design, the bioreactor type that I decide to use directly impacts the form of the scaffold that can be used. So now looking at some of the actual types of bioreactors, Jan alluded to this. Basically, there are many different bioreactor configurations available, and these have been developed for a range of different cell culture techniques, whether it be fermentation of yeast or bacteria or other mammalian types. However, we now need to determine which bioreactor configuration is best for our cell type or species. And it might be that it's none of these, and we need to go back to the drawing board and completely innovate or build a custom bioreactor. Or we might require a combination of some of these. But essentially what we want to see is a move from the left-hand side, which is your manual lab scale culture, towards more automated systems, which allow us to scale up production and bring down the costs. As I mentioned, my focus is on hollow fiber bioreactors. But within our lab at the University of Bath, the Ellis lab, we also deal with a range of other bioreactor types. And what I want to draw your attention to here is essentially the relative volume difference between some of the automated bioreactor types of stirred tank, fluidized bed, and hollow fiber bioreactor compared to your more manual lab scale culture in tissue culture flask. So you can see that the relative volume starts to decrease. And the reason for this is that you can increase the cell density that can be grown within the reactor. And this is a result of overcoming such limitations such as mass transfer. However, these relative volumes are currently based on an average for general mammalian cells. And what we now need to do is actually work out the cell densities within these reactor systems that are specific to our cell types of either muscle cells or fat cells, and also for different species. But if we're able to do that, there are a number of benefits that we may see. By using a smaller reactor volume, we can reduce costs in terms of capital expenses for purchasing the reactor, and also floor space requirements, and even energy required to actually maintain isothermal conditions. So research within different bioreactor types will contribute greatly towards that acceleration phase of cultured meat as a technology and move us towards a more ideal system. But it's not as easily done as said. So there are a range of considerations when you're thinking about bioreactor design. And then all of these feed further into overall bioprocess design. But for now, I'm just going to take a step back and look at some of the custom technology that we use within our lab for actually producing scaffolds that can be used within our bioreactors. So for some of the bioreactor types that require microcarriers, we have designed and 3D printed a microfluidic device that allows us to produce microcarriers. And in this case, I'm producing microcarriers from silk fibroin, which is isolated from silkworm cocoons. And in the video, you can see the actual microcarriers have been dyed blue and are being produced via the microfluidic device. Now, this is an example of custom technology within our lab. 3D printing is a great resource for producing custom technology. It provides the user with flexibility, potentially producing components with fewer parts. And from a research perspective, it is greatly beneficial to optimization of processes because you can, the speed and turnaround from the point that you design to use is greatly improved. And over here, I just have some microscope images of the microcarriers that have been produced. You can see they are nice and spherical with a small size distribution, which is very important for the fluid dynamics within a bioreactor. And below it, you can see some of my cells stained in green growing on the microcarriers. So these are suitable for bioreactor types such as stirred tanks, fluidized beds, and wave bioreactors. But I mentioned that my work is primarily on hollow fiber bioreactors. So now I'm just going to talk through a bit about the production of those membranes. And when we're deciding on a material for use within the hollow fiber bioreactor, it's much simpler to first test that material in a 2D form. So we produce what we call flat sheet membranes. And then we've got a bit of custom tech where we've taken your common lab equipment of a well plate, and we basically designed and adapted this to basically clamp in our flat sheet membrane so we can just grow our cells on this 2D surface. So quite often, custom technology, you can think of it very simply as just the optimization and adaptation of existing technology. You don't need to completely reinvent the wheel. And I would say that within academia, we are very good at doing this. We make use and make do with what we have available to us at the time. Now, once I've determined that this material is cytocompatible and my cells can attach to, grow on, and remain alive on the material, I then produce the hollow fibers from this. And here we have another bit of custom technology. We have our wet spinning rig, where we basically use a concentric orifice spinneret to produce the fibers via a process known as phase inversion. And below you can see some SEM images of my hollow fibers. And by producing these in-house, it gives us more control over the scaffold properties. We can control things like the wall thickness, the wall porosity, and consequently permeability, and also the dimensions of the fibers. And for those of you who aren't necessarily familiar with hollow fiber bioreactors, this is just to give you a quick introduction to them. The main benefit that allows us to culture the cells at a high cell density is because of the fact that the cells can grow on the outside of the fibers, which means we can decouple them from the shear stress of the flowing liquid, as well as overcome mass transfer limitations and oxygen diffusion limitations. So you can see in the top left-hand image, that is a cross-section of a hollow fiber bioreactor. And in the photo, this is a photo of one of my bioreactor setups. And there in the middle, that is the actual hollow fiber bioreactor. So we are currently taking a scaled down approach where we are looking at a very small system to define some of the fundamental operating conditions for these reactors and our specific cell types before we then scale up or scale out. And another bit of custom tech that we have here is the actual bioreactor module is made of glass, and we actually get these glass blown to meet our design specifications and dimensions. And I think this is a very good example of basically taking advantage of existing skills and knowledge from other fields and other areas of expertise, whether that be outsourcing or collaborating with other teams, or by actually building your own team with a multidisciplinary approach, bringing in people with a wide array of knowledge. And so very much a quick overview of some of the work that I do, but thank you all for listening, and I look forward to the debate.
Thank you very much, Scott. There are a few questions under the Q&A sections popping up for you specifically, so you can type the answers out as we speak now. Okay, thank you very much, everyone, for these very interesting presentations. Now let me kick off with a bit of an insight on that. So a lot of you have mentioned kind of systemic approaches to building efficient bioprocesses and the co-development of technology and bioprocesses themselves. So The question to you, Ivana, as I saw, you have multiple centers that each deal with a specific part of this kind of this whole picture. How do you manage the collaboration between them to make it efficient and quick and productive in a let's say acceptable time scale?
Yeah, that's not that easy, especially since we are growing. So each of these centers have quite a large number of people. So it does happen occasionally that one center, the people in one center don't even know what projects the other center has. But we are trying to have the inter-center groups, like a group which consists of people from different centers. For example, this Center for Biosystems, in which I am and my PhD student Mila, we collaborate a lot with the Center for Sensing Technologies, where Ivana, for example, is. She's also on the webinar. Basically, we group based on the project. So this Real Sense project basically gathers people from both Center for Biosystems and Center for Sensing Technologies. And as Scott mentioned, it's really necessary to have this multidisciplinarity, because it's really difficult to know all of these technologies. Just, you know, I'm a biologist. I focus on the cell culturing, on the type of the cell. But for example, this the microcontrollers for controlling the electronics, for measurements, that's definitely engineering part. So without this collaboration, it wouldn't be possible to do anything. And but to answer directly your question, how we manage, it's challenging. It's difficult talking even with in a multidisciplinary team. It's a challenge to communicate when people are from different backgrounds. So even finding this common language is kind of tricky. But I think during these interdisciplinary, multidisciplinary communications, that during this we actually can reach new findings, because different people, different backgrounds can shed light on different aspects of the same phenomenon. So it's basically just efficient communication.
Yeah, I was kind of thinking in the same direction as well. What we have seen at IRNAS is that exactly what you've mentioned, having a perspective on the same problem from people from different kind of fields of expertise has never resulted, you know, never had adverse effects on anything. So it's in our experience, this sparks up a lot of innovation. But there are challenges along the way, especially in terms of the mutual language and the way of thinking and the workflow kind of what we are all used to. Everyone is used to different things. So maybe Jan, your approach to development of bioreactors is, you know, your core business is having it modular. And then customizing it for the final, you know, for the final application. How do you keep the costs of customization low? What would be your input on that? There we go. Yeah.
Oh yeah. Now you can hear me. So the trick is actually quite simple. Anything that is specific to a module, keep it close to the module. So basically, a typical module is a physical sensor, or it is the actuator, like the motor of a pump or a rocking motion platform or something. And obviously, you need to communicate with that thing, and there's going to be a piece of electronics and a microcontroller that is directly doing that communication. But as soon as we are in the microcontroller world, it's only software, and software scales inherently. So as soon as I am in the software world, it does not matter if my microcontroller controls a tiny motor or a huge motor. And if I need to control many motors or just one motor, because a software module, I can replicate as often as I want. So I sometimes say we're even more a software company than we are a hardware building company. The hardware is just a means to get to something, but the value actually is created in the firmware and in the web application software and in the control system.
Yes, I was imagining something of this in this direction. Thank you for confirming my thoughts. The approach we chose at IRNAS is creating platforms. So you get to some kind of a minimum, let's say a minimum viable stage that you can then kind of jump from and scale it up, or tune it and tweak it and customize it quicker. And that's the way we try and save the money on it, and also keep the cost low, because it allows us to not start from scratch every time, and build on something that's already been developed and validated and verified, and we make sure that it's adopted, with in early stages, with that enough people adopt it so we can really extensively test its functionality and see. And kind of draw the line and say, okay, this is the minimum. We have it right now. It works. It functions. We want to build on that right now.
Yeah. Well,
and
I mean,
it's been surprising to me that a lot of the times the cost of customization of a particular platform is in the same range as buying an off-the-shelf, high-end, or even mid-range piece of equipment. It often people associate customization with a huge number in terms of cash with it, and it's not always the case. I'm interested in what your input is on this.
Yeah, well, I mean, you are right. The point is, if you have a modular system that allows you to just put the stuff together, plug and play like Lego, it doesn't have to cost very much, because we do not have so much development effort that we have to put on the invoice. It's like, if we have a module for it, then you basically get it for the price of an end product. But if you want us to develop a specific new type of vessel, or integrate some strange sensor that the world hasn't seen before, then obviously I need to pay the engineers to do that. But it does not cost very much to just put together something that is meant for putting it together easily. So that can really bring the costs down. And also it brings the costs down for the user, in the sense that I said before, you might not even know in which direction your project will go. So why do you need to spend all the money on the final device if you don't know what the final device is? So start with a simple system, play with it, learn something about it, and then once you have your initial results, start extending it. And this is what a modular platform can also give you. So you need a pump more? Well, just get a pump more and you just attach it to whatever is there. You need another sensor? Well, we'll just add it for you. And that's obviously with some technologies that is already possible, if the maker has foreseen that you have different configuration options, but it stops with the configuration options. And in our case, there's no limit. You can just go on. And that really makes sense, I believe.
Yes, I have the same impression. Maybe what is your well, that's not directed at anyone. It will be interesting to hear thoughts of all of you. But what do you think about because if you have let's say an off-the-shelf piece of technology that's in a serious production, mass manufactured, you can buy it online. Me as a researcher, or someone who's buying that piece of equipment, I'm just going to assume that once I've acquired it, it's up there and it's, you know, it's certified. It's been through all sorts of validation, extensive testing. A lot of the people that are using it, so I inherently trust the results that this piece of technology is going to provide me, be it either a bioreactor, a microscope, or anything I you know, I might be using. How do you achieve this level of, you know, trust for the user with custom pieces, which haven't gone through the extensive line of, you know, testing and validation and sometimes certification even?
Well, maybe I can also answer this because we are making such bioreactors for these types of users. So it's obviously, if you are doing R&D and if you want to have something that nobody else has because you have a specific bioprocess, you have to take into account that you are trying out something new. So there's no guarantee that your biology plays along. I mean, this is what we often tell our customers. Let's do a feasibility study. Let's see if the cells are really like able to do what you think they can do, and if they even don't have an idea how to automate something, then we develop an idea with them, and then we usually do a feasibility study where we really try out, is the cell surviving to be pumped from A to B? Is the cell liking to be stirred like this, or do we have to stir it in a different way? And you can check this out with a very simple setup. It doesn't have to be all that complicated thing in the end, but you can check if that can work in principle. And then you can build on top of that. And that's also the reason why I said before, don't think about, I'm going into the bioreactor shop and I take the nicest thing off the shelf and then I start working with it and everything's going to be fine. No, you still need to be in a driver's seat and understand what you're doing. And your bioreactor maker should be there for you to help you. Like, they should be able to work out the bioprocess with you. So if your bioreactor maker does not understand your bioprocess, then go somewhere else, because they can only help you finding and developing the right equipment if they really can wrap their mind around your bioprocess. And I think that's the key. So collaboration, iterative approach, that really is something that you have to do in that field if you want to develop something new.
Scott, Ivana, maybe any thoughts from your side on that?
Well, yeah, I definitely agree that it's not enough just to purchase the device. You have to work with the company and the provider to customize it, and that's why maybe it's even. I mean, I'm just thinking. I think Jan also mentioned in his presentation that it's probably necessary for this area of cultured meat to have this approach as they have in OSPIN, to actually work with you, with the research lab. So I think it's not going to work as we have for the antibody production or fermentation. You buy a huge bioreactor and then, okay, for service, you can contact the provider, but that's it. So I agree. You have to, I mean, customization is necessary, especially in this new field, which we don't really know all the different aspects, what to do. But I had a question also for Scott about, and I see also majority of the questions are directed to him, and I think because of this tissue engineering aspect, so it's actually not so much about the hardware, it's the most about the actual well, the scaffolds and the bioreactor. So my question for Scott is, how is the upscaling going to work for the hollow fiber bioreactors? I'm always wondering about that.
Sure, happy to answer. But before we move on from the previous question, I would like to just add my 2 pence in there. If we think about culture meat as a nascent technology, it is likely that we are going to have to use some innovative design. And in terms of verifying that and approving it for use, quality assurance, all technology has to start somewhere. So in this case, it's just a case of we're starting at the beginning. And the way that you actually verify a product is through use. If it's through collaboration with an academic institute, you can then work on publishing that, in which case you then go through the rigor of a peer review. And as more people start to actually use a product, you then start to actually show the repeatability and reproducibility of results with it, which is very important for that quality assurance and quality control. And then I guess just to move on to Ivana's question. So in terms of how we can scale up hollow fiber bioreactors. So actually hollow fiber bioreactors are potentially relatively not simple, but there are a lot more options for the scale up compared to other bioreactor types. For example, if you think about a stirred tank bioreactor, as you start increasing the size to overcome the mass transfer limitations, you then need to actually increase the impeller speed. And that doesn't scale linearly. You have the actual angular velocity that you need goes up exponentially. And that's why with adherent cells, there's a limit to the size, the actual volume of the stirred tank reactor that can be used, because you get the impeller traveling at a much faster speed, which creates a lot of localized shear forces that could detach the cells from the actual microcarriers. Whereas for a hollow fiber bioreactor, in terms of size limitations, that limit is typically based on the length of the reactor, because you will get concentration gradients in terms of metabolites and oxygen along the axial length of the reactor. So there might be a limit to the actual length of the reactor that you can use for a given flow rate, as well as potentially like a size limitation in terms of diameter when you start thinking about the number of fibers that you can pack in there. However, because of the high cell density, which reduces the actual volume requirements of the vessel, and consequently the space required, it does become a lot easier to take a scale out approach. And you can start basically numbering up and operating more of these reactors in parallel. And if they're orientated horizontally, they can then almost be stacked on top of each other. And then there is also the option for what we call process intensification. So actually trying to improve on that high cell density by using either smaller fibers or another way of basically overcoming any mass transfer or oxygen diffusion limitations, so that you can use even smaller vessels or produce more cells within the same size.
Thank you, Scott, for this. Maybe one last question that Ivana and Scott can both answer. So to you, Ivana, first, do you think the custom technology is what's gonna make that diagram new and valid?
You mean this mind map by the
Yes.
Good Food Institute? Yes. As I said, I mean, everything is new in cellular agriculture. Okay, we do use a lot of the tissue engineering principles, but it's a totally different approach. I mean, what you're making needs to be edible. So you don't do that in the regenerative medicine, definitely. So everything is different, and it has to be customizable, customized. And I think both Scott and Jan mentioned that it's not really a good approach to use the equipment made and processes made for biomedical field and then adapt them. Maybe in the beginning, yes, but perhaps we need to devise something from the scratch that is really to the point for this use. And I mean, we are in the development phase, so maybe customization is not even the good term, because customization somehow means also something existing and then you customize it to your needs. But maybe we are developing new technologies, new processes.
Mm-hmm. Yes, I agree. I think customization might be a little early for customization.
Maybe.
But anyway, I think that co-development of workflows and processes with technology is the key forward. So Scott, what do you think when you are attempting or thinking about applying some custom piece of technology in your work, how important it is that your current workflow is not disrupted? From what I've experienced before working with researchers, sometimes people tend to like to stick to their old kind of principles and the principles of operation that they're used to, and some are more and some are less kind of flexible to adapt new technology and new approaches and something they've never seen before. What's your experience, and what's your thought on that?
I would say, firstly, it depends. If we're looking primarily at PhD research, a lot of it will come down to the stage the student is in their research. When it's early on, you certainly feel less pressure, and so you feel like you have a bit more freedom to almost play around and try out new things, spend time on different projects. However, as it gets to the end of your funding, it becomes a case of, right, it's now crunch time. I need to focus on what I have been doing, what I know works, and basically getting as many results as possible. What I have found is that quite often what actually leads to this custom tech and innovation is when you encounter a problem that you can't actually overcome either with the technology or the equipment you have available to you within your lab or at the institution, or you have a problem that all the commercially available options are actually just too expensive and outside your current funding availability. And so that itself almost ignites a fire within you and causes you to be a bit more creative and search elsewhere for alternative solutions.
Yes, I agree. I think also because we're, like we just mentioned, we're talking here about a very young kind of piece of like very young field, a very something that's getting its form slowly, slowly becomes unblurred. I think people have to be kind of open-minded enough to admit to themselves that the processes that they are they've been used to up until this point, it might not be enough. And that to get them where they want to get. So not to be discouraged by something new coming in and changing their workflow a little bit, so they can then enjoy the new practicalities and options that new technology unlocks. There are a few questions for everyone under the Q&A. There's one for you, Ivana. There's a few for you, Scott. We are coming to an end of the webinar, so we will make sure all these questions get answered through emails. And what I would like to ask all attendees is we're going to be sharing all panelists' email addresses in the follow-up emails you all are getting. So if you have any particular questions regarding the work of any of the panelists, or you need any specific advice, they will be more than happy to get, feel free to get in touch. They'll be more than happy to help and share their experience with. I hope we've sparked up some ideas today. Hope we've touched on a few topics that people are going to go home and think about and talk about as well with their teams. I think custom technology has a very good potential to be incorporated in this young field, even more so maybe than in any kind of well-established big field. And we're here to take advantage of it. So thank you again for listening. Thank you to all panelists. Enjoy your summer. I wish you a lot of success hopefully your labs will be available very soon, and you can continue your work undisrupted. And yeah, feel free to visit the same place you visited to register for the webinar. That's where the recording will be published, and hope to see you soon.